I'm presenting a paper that we published in Nature Biotech about half a year ago. And the first author, Tomas Rube, is a former postdoc who started his own lab in California. He couldn't make it, so I'm giving the talk on behalf of the whole team. But we're a lab that's... So, transcription factors. We're interested in understanding really quantitatively how DNA-binding transcription factors bind to DNA. And I was trained as a physicist before I defected to biology, so I like to think of functional genomics as a biophysics experiment that happens to include a genome like chromosome molecules, right? So now, if you look at the early history of how weight matrices, right, for representing binding specificity of transcription vectors were defined, it really came from multiple alignment. We saw some talks about multiple alignment, right, which is in an evolutionary context, comparing different sequences, and then you start to count how often you see a particular amino acid at a given position. So these weight matrices that people started using also for protein-DNA interaction were also in the early days because the data came in the form of small number of examples of binding sites, right, in a multiple alignment, and you would start to count A's, C's, and G's and T's, thank you, and try to summarize that in a weight matrix. A small step for mankind, but a giant leap for this presentation. Okay, so we're interested in understanding chromatin very broadly in terms of how these proteins bind to DNA. We're interested in cofactor interactions, also in low affinity binding sites. And there's evidence from papers from the last few years that binding sites that are even 100-fold lower than the best site in the genome can be fully functional. So you need to be able to kind of quantify binding and the impacts of, for instance, non-coding polymorphisms on the binding of transcription factors if you really want to make sense of non-coding variation, for example. Now, about 15 years ago, we introduced this logo representation where the letters are above or below the line rather than bit scores. And the reason was that we wanted to have a logo where the height of the letters corresponds directly to free energy differences associated with making point mutations in the DNA binding site. So the scale here is kcals per mole, or RT if you want, right? Like thermodynamic units. And that means that height of a letter corresponds to a specific fold change in the affinity, corresponding to a specific additive change in the energy. Energy is just a log of a relative affinity, right? So we very much try to keep in mind the structure of these transcription factors and try to come up with a biophysical representation. Now, several decades ago, Gary Stormo and co-workers were trying to do linear regression of these energy parameters to measurements of affinities of reference sequence and mutated sequence, right? Then around this time, my lab started to build models for fluorescence-based DNA microarrays, where now you had this probe on the substrate, there's the protein binding to the probe at different offsets, right? So the predicted intensity of that probe would be a sliding window sum of an energy model, and that energy model itself would be a sum of energy contributions, right? So it would have this form, which is, these days you would say that's a two-layer convolutional neural network. It's the same thing, a sliding biophysical model with a sliding window where you sum up energies within the binding site. It's equivalent to a two-layer neural network, right? So in that sense, it's a lot of the more current algorithms they're kind of deeply related to some of this earlier biophysical work. Now, of course, about 10 years ago, people switched from DNA microarray-based data to deep sequencing data, right? And for protein-DNA interaction in particular, there's a very nice type of experiment you could do. You take random DNA libraries, you synthesize from random oligos, double-stranded DNA molecules, but a huge number of different molecules in a test tube, mix it with a purified transcription factor protein, and then run a gel to separate the bound from the unbound DNA. And then you can build an NGS library from the bound DNA. You also build an NGS library from the input DNA, and then somehow from the evolution of that set of sequences, you hope to infer something about this mapping from sequence to binding affinity. Now, if you think about the nature of this problem, and you can actually do this over multiple rounds, right? If you have a 30 base pair, oops, I'm pressing the wrong button. 30 base pair probes, 4 to the 30 is 10 to the 18, right? So the space of possible sequence that you could sequence, if you sequence one read in your data, is huge, way larger than the number of reads that you sequence. And if you assume that the initial pool is more or less random, then the expected frequency of any particular sequence is about 10 to the minus 12. Right, so you cannot expect to see enrichment and see counts in the hundreds or something and be able to compute a ratio of input versus selected and get any kind of precise measurement of enrichment that maybe then you could relate to a relative affinity. Now, the optimal sequence and the non-specific binding level are typically separated by 1,000-fold or something, right? So again, from a biophysical point of view, there's a huge range of affinities that we want to cover with the algorithms. You could be 1,000-fold above non-specific binding and also 1,000-fold below optimal binding. And then the question is that functional or not? Now, I don't have time to go into the technical details. You can read them in this paper. This first paper, we basically came up with a multinomial formulation of this problem where we say the NGS data, the FASTQ file, is a multinomial sample of size 1 million from this space of 4 to the 30 sequences, right? And the probabilities of the multinomial are related to this biophysical parameter there's in the biophysical model for binding that we have, right? So it's basically a regression problem. You're trying to fit delta G parameters, 3 mutations at every position. And you try to get those numbers directly from these sequencing counts. Well, that was hard, and we have like 80-page method section that's basically Chaitanya's PhD thesis to set up a recursion relationship to compute the denominator of the probability, which is the sum over all possible sequences and their expected probability, right? But we were able to solve this, and the nice thing is that this gave us really powerful predictive power. And again, I just want to go over this quickly, but the kind of validation that we did is we took an enhancer from Drosophila, looked at the stripe pattern in a fly embryo, quantified the intensity of the stripes, and then we showed that we can actually predict the enhancer activity as quantified from this microscopy data even though these individual binding sites in this enhancer are about 300-fold weaker than the bus site in the genome, right? And there's real experimental validation in vivo, but the model that we built, which is over here, I should be using this pointer because it keeps moving back. Is over here, right? This model was built from in vitro data, this random DNA molecules in a test tube and purified to supervector. Okay, so it was surprising that you could do so well on these very weak binding sites. But there were lots of restrictions. It was only applicable to one round of experiment. It couldn't be saturation, etc. And so when Tomas Rube, who was the first author on this paper, joined, he came up with a way to reformulate the problem that made it much more flexible. Now we could write down any kind of biophysical model, arbitrary functional form, number of parameters, and then fit it to this NGS-based functional genomics data. And the first thing we did was actually reanalyze, and this gives an overview of the kind of assays that you can model with this. But the first thing we did is we reanalyzed hundreds or even thousands of in vitro experiments from UC Taipei's lab, Bart Planke's lab, and some other groups, and built these models. And then part of this database, which is called motifcentral.org, you can go there and look up your favorite transcription factor. But for every motif that is there and that you might want to use to score your own sequences, we provide for that motif all the validation data, correlations with fluorescence-based microarray measurements, ROC curves for ChIP-seq, etc. Right? So you can really look into the quality of the motif before you use it and decide whether it's any use. And one particular measure that we assign to the motif is what we call the MAFR, or the meaningful affinity fold range. Can you trust this model over 10-fold range of affinity, or 100-fold range of affinity, or even 1,000-fold range of affinity? Now, in some cases, these models actually work over quite a range of affinity. This is a model for CDCF inferred from, again, in vivo data, test tube, random DNA data. But in spite of that, we can predict allele-specific binding as profiled with ChIP-seq over about a 10,000-fold range. So in this plot, this is affinity. This is the best site in the genome. These are sites that are about 100-fold weaker. These are 10,000-fold weaker than the best site in the genome. And when these points are above the line, it means that we correctly predict which of the two alleles for that SNP is more strongly bound by the transcription factor, right? And the consistency is then in terms of whether the ChIP-seq data told you what the direction is of preferred binding, which allele has the more aligned reads at that position in the genome. But you can also do more fancy things like look at complexes of 3 different homeodomain factors, do these in vitro experiments for each of those proteins at this, separately or jointly and mixed in the same test tube. And then from this data, which is all these different types of combinations of these homeodomains, we can fit a model that models all the protein DNA interfaces between the various subcomplexes, but also actually tells us in energetic terms like kcal per mole how much the stability of the complex increases or decreases if I increase the spacer by 1 base pair, for instance, right? So very detailed, almost structural information, but we don't use structure at all in these experiments. It's purely a sequencing-based biophysical assay, if you like. But using this, this deep learning or type biophysically informed machine learning, we're able to tease out all these mechanisms. By the way, the algorithm we call ProBound, it models whether the protein is bound or what the occupancy is a function of concentration and all that. It also is supposed to sound a bit like profound, which is a wink to deep learning. And it has elements, of course, because of the sliding window, the 2 layers in the neural network. But we didn't use any of the standard machinery there. We just come up with the algorithm from scratch. Okay. You can also do things, and I'm just going to rush over this because in the interest of time, but you can quantify the impact of methylation of a cytosine at any position in the binding site, again in quantitative terms, like how much energy do you gain or lose if you add a methyl group to the cytosine at a particular position. And then also, perhaps surprising to for some people is like you can infer absolute KDs in nanomolar units from this deep sequencing data as long as you sequence not only the input and the bound fraction, but also the free DNA. And again, you're using this gel shift to separate the bound from the unbound DNA, but if you jointly model those 3 libraries, the model can actually predict absolute binding affinity in nanomolar units. And this plot shows a validation of that, where this is for 4 different DNA sequences, the prediction of our model in nanomolar units. And then here is measured KDs from a titration curve of individual DNA molecules, and then with a varying concentration of the transcription factor, and where the inflection point is, that's the KD for that sequence. And the scales here is logarithmic, so this is again over, in this case, like about 100-fold range of affinity. Are these predictions correct? Maybe interesting for some of you, we also found a way to do peak-free motif discovery from ChIP-seq data. So, of course, the standard pipeline is you look for peaks and then you look for motifs enriched under the peaks. But looking for peaks is kind of equivalent to looking at the extremes of the distribution, and in a way you're losing the quantitative relationship between binding, biophysical binding, and the probability of observing the read in the data. So instead what we did is we basically treat the input and the immunoprecipitated samples in the IP experiment more or less as if it was the input and the output of this in vitro selection experiment with the random DNA molecules, right? And when you do that, you get models that are very similar to the models that were derived from the in vitro data. So just to make that implicit, here at the bottom is CellX or SMILE-seq data from Bart de Planke's lab. In vitro data, random DNA molecules, purified transcription factor, right? And this is our model. These are delta Gs in kcal per mole, or RT units in this case, right? And then the plot over here on the right is a direct comparison between the delta Gs from the ChIP-seq data on the y-axis and the in vitro binding data on the x-axis. So it's very close to a straight line. It's R squared of 96% over, again, like 100-fold range of affinity. So it means that ChIP data can actually give you highly quantitative binding energy models. And we actually only used 100,000 reads from the data set. So we subsampled the data to only 100,000 reads. It wouldn't be enough to call even a single peak because the genome is 3 times 10 to the 9, right? But in spite of that, just from the statistical pattern of how these reads distribute over the genome, we're able to infer this energy model that's completely consistent with the in vitro model. But then, of course, ChIP-seq data, you also get additional motifs. We can tell our algorithm to look for additional contributions to the selection. And then we found 3 motifs that each correspond to known cofactors of the glucocorticoid receptor. In this case, is a steroid hormone receptor. And so this is one ChIP data. We're of course very interested in applying this more systematically to say all of ENCODE and try to see if those models that implicitly take into account some of the cell type dependence, because these cofactors, they are cell type specific, whether that, for instance, gives us improved ability to predict allele-specific binding, right, on the non-coding SNPs. The final application was actually going from protein-DNA interactions, and we also did this for lots of RNA binding proteins, by the way, but in this last example, we used peptide display. So you can actually take E. coli bacteria, transfect them with a plasmid library that makes them display a random peptide on the outside of the cell. So there's some cell wall protein that you add a little N-terminal part to it, right? And then when you expose those bacteria to a kinase, in this case, the tyrosine kinase SARC will start to phosphorylate these tyrosines in these little peptides, right? Every bacteria is covered with a different kind of peptide. So from these data, which is a time course actually, we build a model that now, instead of predicting KD from the DNA sequence, was predicting the enzymatic efficiency, like how fast is a particular substrate sequence being phosphorylated by the tyrosine kinase, right, as a function of the peptide sequence. And in this case, we also did an experimental validation where on the x-axis, it's the predicted enzymatic efficiency in like per second per micromolar of the enzyme, right? And then on this axis, we actually synthesized individual peptides, and then it's HPLC, which is high-performance liquid chromatography, to measure the degree of phosphorylation, right? And this is over, again, like a 10,000-fold range, but if these pairs of points are actually non-synonymous SNPs, like 2 alleles of some peptide substrate of SARC, right? And if these lines are parallel to the diagonal, it means that the model predicts the full impact on enzymatic efficiency accurately. If they're all aligned, it means that the model is actually even predicting absolute rate. But this is a hard problem, right? So again, you can also use this kind of approach using protein display or peptide display to look at protein-protein interactions, in this case, enzymatic interaction. Okay, so with that, I'd like to wrap up again. Shout out to Tomas, who actually worked very closely with Chaitanya, who's the first author on the first paper, and then other people in my lab who were either generating experimental data, Judith Kribelbauer, who's now a postdoc in the lab of Bart de Planck at EPFL, and also Richard Mann, a collaborator at Columbia, and his postdoc Siqian on what we call KD-seq, this absolute affinity inference, and Neel Shah and his grad student Allyson on the SARC protein-protein interactions. Okay, so with that, thank you for your attention and for staying all the way till this last session.